Hello, my name is Dr. Ominde, and I'm going to discuss the histology of the female reproductive system. So, this comprises the ovaries, fallopian tube, uterus, vagina, and the external genitalia. And the main function of the female reproductive system is to produce gametes, that's the exocrine function of the ovaries, to produce sex hormone through the endocrine function of the ovaries, transportation of the egg by the fallopian tube, again the site of fertilization at the ampulla fallopian tube, and after fertilization the oviduct is also able to transport the zygote to the site of implantation. The uterus serves as a site of fetal development, while the vagina is a copulatory organ and the birth canal. The ovaries are usually ovoid and they are covered by a surface lining epithelium, which is the germinal epithelium. It's simple cuboidal epithelium. Below the epithelium is connective tissue, which we call tunica albuginea, and after connective tissue, the parenchyma of the ovary is divided into an outer cortex and an inner medulla. The outer cortex contains ovarian follicles at various stages of maturation, up to the level where you have corpora lutea and the corpus albicans. These follicles are embedded in loose connective tissue stroma. The medulla of the ovary contains connective tissue and blood vessels, and the medulla is usually continuous to the mesovarium, which is the peritoneal membrane around the ovaries. The process of folliculogenesis is basically maturation of primordial follicle. So from primordial follicle to a mature follicle, and this occurs throughout the reproductive life. The follicle is an oocyte that is surrounded by epithelial cells. Folliculogenesis, where the follicular growth and maturation is under the control of follicular stimulating hormone produced by the anterior pituitary. We also have luteinizing hormone that is responsible for regulation of estrogen synthesis and also for ovulation, whereby a surge of LH occurs 24 hours before ovulation. So this image just shows you the ovarian cycle here and the uterine cycle. So our main focus is the ovarian cycle. You can see the primordial follicle. It starts maturing during the bleeding phase or the menstrual phase of the uterus. So the ovarian cycle is divided into three: follicular stage, ovulation, and luteal stage. So follicular stage, you mature the follicle up to the Graafian follicle, and when the fluid builds up, it pushes the ovum out. That is at day 14, and that occurs. This is the process of ovulation, and this occurs 24 hours after the luteinizing hormone surge. So this ovulation corresponds to the proliferative phase of the uterus. So after shedding, which is bleeding, the uterine wall starts to build up. It thickens up. Uterine glands begin to form and accumulate in mucus and glycogen. So after ovulation, what happens to the remaining follicle after it has released the oocyte and its corona radiata? It becomes corpus luteum. And corpus luteum is able to produce progesterone that is able to cause the uterine wall to even grow more, make the uterine glands to be more tubular and more tortuous in preparation for fertilization and preparation for implantation of the fertilized ovum. But when fertilization does not occur, what happens to corpus luteum? There is no need to prepare for pregnancy, so corpus luteum degenerates because progesterone is no longer needed, and it becomes fibrosed as corpus albicans. So the primordial follicle is just a primary oocyte that is lined by simple squamous follicular cells, and this primordial follicle contains oogonia, which come from the yolk sac, and the oogonia undergo fast meiotic division. So from oogonia, you're able to form the primary oocyte. Now, this fast meiotic division is usually arrested at prophase, and this takes very long. Stromal cells usually form a layer of follicular cells on the basal lamina around the primary oocyte. The primordial follicles are found at the outer portion of the cortex, and this is just below the connective tissue, which is the tunica albuginea. So we have two types of primary follicle: the unilaminar, that has one layer, simple cuboidal epithelium of the follicular cells. Then we have multilaminar primary follicles, which is a primary oocyte enclosed by several layers of follicular cells. So follicular cells proliferate by mitosis and form granulosa cells. These granulosa cells have receptors for follicle-stimulating hormone. We have zona pellucida, which is a glycoprotein coat between the oocyte and the granulosa cell. It's gel-like and homogeneous and acellular, contains neutral proteins that are produced by the granulosa cells and oocyte. So both the granulosa cells from the follicular cells and the oocyte produce this glycoprotein coat that we're calling zona pellucida. Okay, so we have microvilli from the oocyte penetrating the zona, and this contacts the microvilli of the granulosa cells. Therefore, there is communication via gap junction. So in as much as we have zona pellucida separating the oocyte from the granulosa cells, these two cells communicate via gap junctions because the microvilli from the oocytes are able to penetrate the zona and communicate with the microvilli from the granulosa cells. So this is your oocyte, and this is the glycoprotein called the zona pellucida, and these are the granulosa cells of the follicular cells. So follicular stimulating hormone causes the surrounding stromal cells to differentiate into layers of thicker cells, and these thicker cells are large and pale staining, and mainly responsible for steroid synthesis. So these layers are highly vascularized. So the stromal layers differentiate into thicker cells, which are steroid synthesizing cells. They are large and pale staining and highly vascularized. So after you formed your primary follicle, you get your secondary or antral follicles. The follicles start forming liquid-filled areas between granulosa cells. These liquid-filled areas coalesce and form an antrum, which contain liquor folliculi. The antrum enlarges and pushes the oocyte to one side of the follicle. Therefore, the oocyte is now surrounded by a layer of follicular cells. These are the corona radiata, and the oocyte will rest on granulosa cells that we call the cumulus or forus. The granulosa cells are protein and steroid synthesizing cells. They are able to convert androgens produced by thicker internal cells into estrogen by aromatase enzyme. The thicker differentiates into an inner thicker interna and the outer thicker externa. Remember, thicker interna causes steroid synthesis, while thicker externa is just connective tissue. So granulosa cells carry out protein and steroid synthesis. They contain aromatase, where they convert androgens into estrogen, and this occurs by the thicker internal, mainly for steroid synthesis. The thicker externa is mainly connective tissue. So you need to remember the process of oogenesis, where you have an oogonium that undergoes mitosis to form primary oocyte. This primary oocyte undergoes meiosis 1, but arrest will occur at prophase 1, okay? And this arrest continues throughout childhood up to puberty, when ovulation begins. So ovulation induces the completion of primary meiosis, and secondary oocyte will be formed. So this will begin secondary meiosis that will be arrested in metaphase. So when do we complete this second meiosis, or this meiosis 2? Meiosis 2 will be completed when this oocyte is fertilized by a sperm. So fertilization causes the completion of meiosis 2. Remember, after you form secondary oocyte, meiosis 2 begins, but it is arrested at metaphase 2. So after fertilization, you complete the process of meiosis 2. So oogonium undergoes mitosis to primary oocyte. Primary oocyte begins meiosis 1, but it will be arrested in prophase 1, and all this occur before birth. So this arrest continues up to puberty when ovulation occurs. Meiosis 1 will be completed. You form secondary oocyte. Meiosis 2 begins, but it is arrested at metaphase 2 until when a sperm is available to fertilize the egg. And when fertilization occurs, meiosis 2 is completed. And here it just shows you the follicular cells around the oocyte, from primordial follicle to primary follicle to a growing follicle, then liquid filled areas with liquor folliculi to become Graafian follicle, and then ovulation that induces completion of meiosis 1 and beginning of meiosis 2 that is arrested at metaphase awaiting fertilization. So this is your primary oocyte within a growing follicle. Then you start forming spaces that are filled with fluid. You have your antrum there, corona radiata cells surrounding the oocyte, oocyte separated from granulosa cells by zona pellucida, and the oocyte resting on granulosa cells, which you call the cumulus ophorus. And you can appreciate here fertilization that will cause completion of meiosis 2. So a mature Graafian follicle as the oocyte with its corona radiata, the oocyte with its corona radiata cells will detach from the cumulus ophorus. Then it's now freely floating within the follicular fluid. So the follicular cells now will acquire a LH receptor, and this is critical for the development of corpus luteum later. So you detach at the cumulus ophorus. This now floats freely, awaiting ovulation. And these are the thicker internal cells that carry out steroid synthesis, thicker external cells that are connective tissue cells. Okay, this is corona radiata, this is cumulus ophorus. So what happens during ovulation? The Graafian follicle reaches its full size. There's rapid accumulation of fluid without significant increase in pressure. The follicle then bulges at the ovarian surface to form a stigma. So the small oval area called macula pellucida or stigma appears where the follicle is bulging on the ovarian surface. This area will change in color, translucency, and integrity. And when estrogen now reaches the maximum level just before ovulation, there will be an LH surge to cause ovulation. So this ovulation is where the follicle ruptures and there's a sudden release of follicular fluid as well as the ovum, which is usually surrounded by corona radiata. So the corona radiata cells will disperse in the presence of spermatozoa because during fertilization we have acrosomal enzymes that will cause dispersion of the corona radiata cells. The fimbriae of the fallopian tube during ovulation will now pick up the ovum, and if the ovum eventually is fertilized, a zygote is formed and begins cleavage, and then after that it goes to the uterus for implantation at the upper posterior wall of the uterus. If the ovum is not fertilized, it will degenerate in 24 hours because if it's not fertilized, it degenerates in 24 hours. So we have maturation of the oocyte, yolk sac endoderm produces the oogonia, okay, which are primordial germ cells, and they will undergo mitosis to form primary oocyte. Primary oocyte will start meiosis 1 during fetal development. This will be arrested in prophase 1 until puberty. The primary oocyte is usually incorporated into a primordial follicle. Meiosis 1 during ovulation, meiosis 1 that was arrested in prophase will resume, and therefore you form a secondary oocyte. This secondary oocyte immediately starts second meiosis, and this will be arrested at metaphase 2. Now, if the ovum is fertilized, meiosis 2 is able to be completed, but if no fertilization occurs, the secondary oocyte degenerates. So we have some follicles that undergo atresia. So millions will be formed during birth, but small number of follicles reach maturity and are released through ovulation. Majority will degenerate at any stage of development, and this is through apoptosis. So the oocyte, zona pellucida, and follicular cells that degenerate, they are resolved, and basal lamina of granulosa cell remains hyalinized and forms a glassy membrane, and eventually it disappears too. Okay, so that's what you call follicular atresia.